In this video, let's understand the difference between prokaryotic and eukaryotic replication. DNA replication is a process by which two identical replicas of a DNA are obtained from the original DNA strand. The DNA replicates before the cell division occurs. Both prokaryotic and eukaryotic DNA replicate in a semi-conservative manner. However, there are few differences between the prokaryotic and the eukaryotic DNA replication process based on their size and complexity in the genetic material. So let's see those differences. Prokaryotic replication is a continuous process, whereas eukaryotic is a process that occurs in the S phase of cell cycle. Prokaryotic includes cellular and double-stranded DNA, whereas eukaryotic replication includes linear and double-stranded DNA with the end. The DNA replicates in the cytoplasm in a prokaryotic cell, whereas the DNA replicates in the nucleus in a eukaryotic cell. There is a single origin of replication in prokaryotic cell, whereas multiple origins of replication are seen in eukaryotic cell. Small amount of DNA undergoes the replication process in prokaryotic cell, whereas the DNA is 50 times more than the prokaryotic DNA. DNA polymerase 1 and polymerase 3 are involved, whereas DNA polymerase alpha, beta and eta are involved. The replication involves a large Okazaki fragments, whereas small Okazaki fragments are seen in eukaryotic replication. The prokaryotic replication process is rapid, that is 2000 base pairs per second, whereas eukaryotic replication is slow, that is 100 base per second. Two circular chromosomes are obtained in a prokaryotic replication, whereas two sister chromatids are obtained in a eukaryotic replication. DNA gyrase is required in a prokaryotic replication, whereas it is not required in a eukaryotic replication.